Okay, so now how are we going to do this? Similar to the southern blot, we do have the gel. Can you see that this is our gel? We place a membrane onto the gel. Similar. So this membrane is called PVDF membrane. Okay, or if you forget in the exam, just say membrane. So we just place a membrane onto the gel, and we can use a blotter. Okay, blotter is a machine that helps us to transfer. If no, then we just let it like capillary reactions. Okay, so this is a blotter. We can use a blotter. Blotter basically you have the cathodes and anodes. So the migrations, can you see that? So from the positive end to the from negative end to the positive end. So that's why it moves. Okay, then it will be trapped inside. Again, we trap inside this membrane. Okay, so when it trapped inside this membrane, what we're going to do here is we're going to use a monoclonal antibody. So how it look like? Okay, look at this, the process. So first thing, this is my gel. So the gel will contain the protein that we want. Okay, different type of protein we separate. Okay, let's say this part is a negative terminal, this one is a positive terminal. So the migration, okay, directions will be from negative to positive because we code them with the SDS and we code them with the SDS. So what we're going to do here is we place a membrane. And then we can place the blotting paper as usual. If we are rich, then we can have the blotter using electric it's a field to transfer. So when it transfers, some of this protein will bind to this, will be trapped onto this PVDF membranes. Okay, this is a PVDF membrane. So in the exam, if you forget, just write membrane enough already. Okay, so what we're going to do here is now, if you view it, you have the membranes already. So this membrane actually trapped with the protein. Okay, trapped with the proteins, a separated protein. Now, but I don't want to detect all the proteins. I want to detect, let's say, for example, the presence of the protein A here. Okay, what we're going to do here is very simple. We're going to find, we can go to the company and buy the monoclonal antibody that target protein A. So two ways we have we have these two ways. One, we just use a primary antibody. So primary antibody basically directly attach to the proteins, a target protein that we want. So this is a monoclonal antibody. So when a monoclonal antibody binds, so this monoclonal antibody carry the fluorescent. It can carry the radioactive substances or radioisotope, or it may carry the fluorescent dye. Can I see that? Okay, so then what we're going to do here is we wash away the unbound radioactive, unbound monoclonal antibody. We wash away these any debris or anything. Then we can expose it. Let's say, for example, if my monoclonal antibody carry the radioisotope, then we can use X-ray to develop this X-ray to develop the image on the X-ray. Then we can view it. Okay, by radioactivity. Okay, so this is a one-step process. But in research, we normally use a two-step process. Why I mean use two-step process? Because we want to save costs. Okay, each antibody is very, very expensive. If you want to target it or tag it with the fluorescent dye, even more expensive. So we use a method number two. Method number two actually save more cost when you deal with multiple protein. Are you clear? We want to deal with multiple protein because if each antibody we want to target or tag it with a fluorescent dye is very, very expensive. So what we're going to do here is very simple. If let's say I want to target two protein, so protein number one and protein number two, you want to check these two protein. What we're going to do here is I have the monoclonal antibody that targets, okay, sorry, use by the same color. Okay, so first monoclonal antibody targets, and then I have a second monoclonal antibody that targets the blue protein. Okay, but both of them, they are not carry any radioactivity. So this one, the first level here, we call them as a primary antibody. And when I buy this primary antibody, I have to make sure I buy from same company, they develop using the same way, and the source, let's say, for example, the source from the mouse. Okay, the source from the mouse. So mouse anti-humans protein antibody, for example, 2. Then what we're going to do here is we're going to have the secondary antibody. So secondary antibody going to bind to the FC region. Can you see that? And then the secondary antibody carry the dye. So this is a secondary antibody. So this secondary antibody, we can use a different source. For example, we can get it from rabbit. So this rabbit antibody target the mouse FC region antibody. So I can view, can you see that? So instead of I have 2 antibody that tag with the fluorescent dye is much more expensive, now I can use this way. So it can apply to all kind of antibody that we use, right? So as long as they come from a mouse, I can use the rabbit's antibody to target, okay? So in our syllabus, explanation is very simple. First, we just say that using the Western blot, we transfer the proteins from the gel onto the membrane. Second, we just say that we can use the antibody. So this antibody actually tag with either talk about radio isotope or we talk about frozen dye. Okay, so it depends on the method. If you talk about radioisotope, then we expose it with the X-ray film. Then you develop the image. If you talk about the fluorescent dye, then we use UV. Okay, use UV. I shine the UV onto my membrane, and then I can capture the image, okay, of fluorescent signal by using the digital camera. Okay, so just these three steps. Enough already. You do not need to go into a very detail. This is for information only. We have 2 ways. Either we can use a direct or indirect. Okay. So monoclonal antibody. There's a use of monoclonal antibody in research. If you need, you can take a photo of this slides or this diagram, and then we go back to our notes. Okay. Okay. So now look at this here. So how we visualize the band by using a Western blot? First, use the SBS page, means for electrophoresis already. We place a PVDF membrane onto the gels, and then we separate the to which the separate protein is gel. In the gel, electrophorically transfer, means that we use electricity to transfer. If we are not rich enough, we don't have a device, then we just use a normal method for the blotting paper. So for you guys, just say transfer. Okay. So next what we're going to do here is the membrane with the transfer protein is then probed with a primary antibody, so specific for the target protein. We wash and remove those unbound antibody, and we can react with a secondary antibody conjugated with the enzyme, for example, Hordish peroxidase, or we can say that it's with the radio isotope or fluorescent dye, okay, in whichever method. So the bound enzyme activity is used to detect the target protein and visualized by a chemiluminescence or chromogenics method. Okay. Now, we're done for the parts that you need to know in your syllabus. Now I'm going to come into this part, what I call isoelectric focusing. Okay, so this is not in the syllabus, but the concepts are slightly different because just now we say that the proteins, right, we separate them based on the molecular weight, but sometimes mutation take place. There is a change in one single amino acid. If you use a normal way to separate the protein, what will happen here is you may not be able to separate them. For example, okay, beta globin. The normal beta globin at the 6 positions is glutamic acid. Correct enough. But the mutated one at the 6 positions now change to valine. And both of them is just a change in one amino acid. It won't cause the very dramatic change in the whole structure. Means the molecular weight. Are you clear? So it means the migration rate may be the same. You may not be able to separate these two, whether it's a normal or mutated one. Are you clear? Okay, so we have to use another method called isoelectric focusing. Okay, isoelectric focusing. So what is mean isoelectric focusing here? Okay, so the protein contains many ionizable group on the side chain R group of the amino acid as well as the N terminus and C terminus. So we have 20 common amino acids. 5 have a side chain which can change, we can be charged. Okay, at pH 7, we have 2 negatively charged R group, aspartic acid and glutamic acid. So they are acidic side chain. Okay, so acidic side chain always give us the negative charge when ionized. And 3 positively charged R group, lysine, arginines, and histidine, with a basic side chain R group. Okay, so now I'm going to show you guys what actually happened to these amino acids when they are charged. Okay, later. So what will happen here is, now look at this. Okay, so look at these amino acids. Amino acid. So central carbon C. So with H and the R group. And then here C, O, OH, and H2. So we learn in the AS, what we know here is in the aqueous medium. In the aqueous medium, what will happen here is ionization take place. So the ionization take place make these, okay, amino acids now become like this. COO, the H ionized, so it detach out. So that's why COO now is a negatively charged with the H plus, detach in the aqueous medium. And what happened at the amino acid site here? And H2, so this H plus can actually attach here to form the dative bond. So in this case, it becomes NH3. Okay, so this H can bind and positively charge. But you can see that overall, this amino acid is still neutral. Can I see that? Overall, even though one side, if you look at this side are negatively charged, this side is positively charged. But overall molecules, they are still neutral. So the one that determines the net charge of this amino acid depends on the R group. So if you revise back for the R group can be positively charged, can be negatively charged, it can be polar, okay, or it can be a non-polar hydrophobic R group. So today our concern is on these 5 amino acids which are positively charged and negatively charged. So how they can be a positively charged or negative charge depends on the functional group they carry. So if my R group carries COOH, under neutral conditions, what will happen here is ionization take place. So this R group now will be negatively charged. Why negatively charged? Oh, sorry, negatively charged because they donate the proton. So because they can donate the protons, so this R group also is termed as the acidic R group. Can you see that? Acidic R group under normal condition, pH 7. Okay, under this neutral pH, then you can see that they are negatively charged. So if I have this basic R group, so what's a basic R group? They have the ability to accept the proton. So you can see R and H2. So at pH 7, also then ionization take place, or they accept the protons in this case, so protons can attach. So you can see that now they are positively charged and they can accept the protons. So this is the basic R group. Are you clear? The acidic and the basic R group. So these 2 types of the amino acids, okay, or these 2 kinds of amino acids, they can determine the net charge of the protein. So it depends on how many acidic R group and basic R group, then you can see that the proteins, they have the final charge of the positive, negative, or no net charge. So how this can be? So at pH 7, what will happen here at pH 7? What will happen here is, if let's say I have 5 amino acid, 1, 2, 3, 4, 5. 5 amino acid. If let's say this amino acid positively charged, negatively charged, positively charged, and no net charge, 0. So this amino acid at the end will be positively charged. Can you see that? At the end, they will be positively charged. Okay, at pH 7. So vice versa, if let's say I have this polypeptide with more negatively charged. Okay, no charge. So this amino acid will be, this polypeptide will be negatively charged. Get clear or not? Okay, so what's mean no net charge? So that's why we cannot say no charge, because confirm they will have the R group with the charge. They still have the charge, but the charge is equal between the positive and negative. Say negative, positive, negative, positive, and 0. Can I see that? So in this case, there are no net charge. Are you clear? There are no net charge. You cannot say, so we cannot, claim that they have no charge, because definitely they have the R groups. Okay, and this positive and negative charge R group can be modified. Are you clear? The charge can be modified by changing the pH. Are you clear? As long as I change the pH, they will change their charge. So that's why the pH, deviation of pH can denature the protein. So how the deviation of the pH denature the protein? Are you clear? The pH. So look at this. If you look at the positively charged R group, COO minus. Are you clear? RCOO minus. So they are negatively charged R group initially. Okay, at pH 7. So if I decrease the pH, how we decrease the pH? We add acid, right? We add H+. So it means that we have decreased the pH from pH 7 to pH 6. What will happen here is we add H+. When we add H+, this H+ now, because extra H+, what will happen? This H+ actually can come and combine with this O minus, because one negative, one positive, right? Reaction actually take place. So can you see that now it can be changed from negatively charged R group, become neutral. Can you see that? At pH 7 and pH 6, there is a change. So because I put in the H+, when I put in the H+, what will happen here is if from negative charge R group become neutral. So it means that the overall charge of the polypeptide can be changed. Are you clear? So once we change, what is the effect? When you change the pH, it causes this. Can you see that if they are negatively charged at pH 7, so because the negatively charged, so means that they can move towards the positive terminal if we carry out gel electrophoresis. You realize that? Because they are negatively charged, right? They are attracted by the positive terminal. So it means that this amino acid or this protein now can move to the positive terminal. Okay? But now if you change it to pH 6, now become neutral, no net charge. When no net charge, they won't be influenced by the electric field. So they won't move, they will stop. So when they stop at the particular regions, then we say that they reach their isoelectric point, where the electricity or electric field won't make them move. They won't influence the movement of the polypeptide. Okay, so now look at another example. So it's R. This is a positively charged R group. Okay, this is a positively charged R group. So at pH 7. So if I increase the pH, So pH, let's say for example, from pH 7 to pH 8. So how we increase the pH? We add sodium hydroxide, for example. So we add OH minus. So what will happen here is, if I add OH minus, this positively charged proton can detach and combine with the OH minus. Can you see that? It can detach and bind to the OH to form water, neutralization process. Correct or not? Okay, so again, now it become R and H2, neutral. When it's neutral already, can you see that? When it's neutral already, again, now they don't have a net charge or no net charge here. So electric field won't have influence. Okay, no net charge, electric field won't have the influence on that. Okay, so for every single protein, so they will have this pi, or what we call this isoelectric point. So what is mean isoelectric point? Isoelectric point actually is equal to the pH where my polypeptide or protein has no net charge. Are you clear? So the protein, they have no net charge. Are you clear? Okay, understand the concept? So how we can do this? How are we going to do this? Okay, now look at this. So if the pH, okay, if the pH is not equal to pi, so means that protein will going to have net charge. Correct or not? Okay, can I see that? When the pH is not equal to pi, so means that protein is going to have net charge. So this net charge can be either positive or negative. Depends on the amino acids. What kind of amino acids the protein has. Okay, so now look at this. I'm going to show you guys 3 types of proteins. Okay, 3 types of proteins. So let's say protein number 1, blue color. So the pi equal to 5. Okay, pi equal to 5. So means that at pH 5, if at pH 5, this protein actually have no net charge. Are you clear? No net charge. Okay, so now as a second protein, green color protein, pi equal to 7. Okay, and then the third protein, orange color, which pi equal to 8, for example. Okay, so understand blue color protein at pH 5, no net charge. For green and orange protein, they have no net charge at pH 7 and pH 8. Okay, or pH 9. Okay. Okay, respectively. Okay, so if these 3 proteins mix together, then you can see that at different pH, they will stop movement. Are you clear? They'll stop movement. So let us look at the first protein, blue color protein. So pi equal to 5. So means that pi equal to 5 basically means that they don't have net charge at pH 5. Okay, so at pH 5, no net charge. But how about if I have it in pH 4? What happened if pH 4? What will happen? Now look at this. If pi equal to 5, pH 4 means we have extra H plus, correct? Okay, they have extra H plus. So it means that with the extra H plus, what we're going to do here is, you can see that now the protein, let me show you. So now, okay, so at pH equal to 5, okay, let me show you. Okay, I start at pH 7 first. Okay, better. pH 7. So means at pH 7, look at this protein. So let's say they have positive charge, they have negative charge, positive charge, negative charge. Okay, and then they have positive charge, for example. Okay, so they have 3 positively charged. Negative charge first, sorry. Okay. Okay, so they are negatively charged here. Are you clear? They are negatively charged at pH 7. So what will happen here? Okay, okay, what will happen here? Look at this. If I decrease the pH from pH 7 to pH 6, what will happen? We add H plus. So what will happen is H plus? I see that H plus will bind to the COOH, correct? Can you see that? Do you realize that? Okay, so means that when H plus bind, so this part become neutral already. Can I see that? It become neutral. So the protein at pH 6, okay, from pH 7 to pH 6, what will happen here is from negatively charged. Okay, original negative charge, right? Now, can you see that? This is the positive, here's the negative, here's the positive, here negative. Now, this part become no charge. Can you see that? If I decrease the pH, do you realize that? If I decrease the pH, so what will happen now? This is no net charge. Okay, so if I continue to decrease the pH, from pH 5, so how we make the pH lower? We add H plus. So we add more H plus. So what will happen to this H plus? This H plus now bind here. Can you see that? When it bind here, what will happen? Look at this, at pH 5. So this is positive charge. This is negatively charged. Positively charged, negatively charged. No net charge already, right? So because of this H plus bind, can you see that now this one original negatively charged now become no net charge? Can I see that? So overall now it become positively charged now. Can you see that? It become positively charged. Okay, so which one is a PI? If you look at this, which one is PI? So PI equal to 6. Okay, PI equal to 6 because no net charge. Isoelectric point now focus at pH 6. Understand? Okay, so means that if the pH, if you look at these diagrams, if the pH, understand, if pH is lower than PI, can you see that? pH lower than the PI, so your protein tends to be positively charged. Are you clear? Understand this concept now based on this diagram? Okay, if pH lower than PI, then it will be positively charged. Understand? So, or vice versa, if the pH higher than PI, can you see that pH higher than PI, it will be negatively charged. So means that once they have the charge, they will be influenced by the electric field. They will influenced by electric field. Okay, so how we carry out this separation, okay, for these 3 protein. Okay, so we are going to have a gel, but this gel is not a normal gel. This gel actually with a different pH. Now, we can make it a gradient. At each regions, they have different pH. Let's say this is pH 2, pH 3, 4, 5, 6, 7, 8, 9, and 10. Okay, different pH. Are you clear? Then we connect lower pH to the positive terminal, higher pH with a negative terminal. And we load these 3 proteins now. Blue protein, a mixture with the green protein, a mixture with the orange protein. So we want to separate. What will happen here at pH 2? At pH 2, for 3 proteins, for 3 of proteins here, blue protein, green protein, and orange protein. you realize that all of them, the pi is lower than pH, correct? So the pH is lower than pi. Can I see that? When the pH is lower than pi, they are positively charged. Okay, so positively charged, positive, positive. So what will happen? All of them now migrate, continue to migrate. Why? Because they are positively charged, 3 of them. So the pH of the gel lower than their pi, so all of them, they are positively charged. Can you see that? So they continue to migrate. So continue to migrate, migrate, migrate. Now come to pH 5. They migrate until pH 5 already. Okay, what will happen at pH 5? Check here, guys. At pH 5, now blue protein, green protein, and orange protein. Blue protein, pH equal to pi. So means that blue protein now no net charge. Can you see that? But for these two, green protein and orange protein, what will happen? Because their pi is 7, pi 9 respectively, so means that the pH of the gel is still lower than pi, they are still positively charged. So if positively charged means that this green protein will continue to migrate, orange protein continue to migrate, but pH equal to pi, they reach the isoelectric point. So basically blue protein now stay. Blue protein now stay because no net charge already. When they are no net charge, so it means that they won't be able to be influenced by the electric field. So continue to move. So continue to move until pH 7. So it means that now you can see that no more green protein, no more orange protein already. So they continue to move until here. So what will happen here? So can you see that green protein now reach the isoelectric point? Green protein will stop, but orange protein, because the pi is 9, pH is 7, still lower than pi. So orange proteins now continue to move until it focus at the pH 9. So this is what we call the isoelectric focusing. So a bit confused, definitely, because it's a bit quite chemistry. But generally, what you need to know here is very simple. Each protein, okay, understand? Each proteins, they have their isoelectric point. So when they reach the isoelectric point, they tend to stop movement, no move, because they don't have net charge. It won't be influenced. So each protein, they have their isoelectric point. We can use isoelectric point to separate them. Are you clear? Particularly for those protein, try to imagine just now we say about glutamic acid, which is basic glutamic acid. Okay, glutamic acid, sorry, they are acidic because they donate H+, they are negatively charged. But if you mutation take place, you change the glutamic acid into the valent non-polar, definitely the isoelectric point will be different. Okay, so go back to our notes here. So negatively charged R groups is acidic. So at high pH, means higher than their pK. So the carboxylic side change lose the H+ ions, so they are tends to be negatively charged. Okay. So positively charged R groups, can you see that they accept proton? When accept protons or these H+ ions, they tends to be a positively charged. Okay, to determine the net charge, we need to know the amino acid sequence and the charge on each side change R groups. Okay, so you can try these two questions later. Okay, based on the given informations, aspartic acid, glutamic acids, they are acidic. So it tends to have a negatively charged. So positively charged with lysine, arginines, and histidines. Okay. So you can see that in this case. So if you look at this, glutamic acid, GLU, glutamic acid, ASP, aspartic acid, ASP, aspartic acid. So these three tends to have a negatively charged R group. Can you see that? Okay, then we have the alanines. Can you see that? We have the lysine. So these two are positively charged R group. So overall, you can see that overall, okay, so this polypeptide at pH 7, they are negatively charged. You realize that? Okay, it's based on the sequence. So how about this? Okay, alanines. Okay, so positively charged. Arginines, positively charged. Arginines, they are positively charged. Okay, and then you can see that negatively charged R groups, glutamic acid only here. Okay, negatively charged R group, glutamic acids only. So overall, you can see that this will be positively charged. Clear? Now, you may see that, how come this one of positive charge, negative charge, actually you balance already. That way, positive charge, negative charge, they are balanced already. Clear? So the net charge on the proteins can be altered. It can be altered, so it becomes more positively or can be negatively charged due to the gain and loss of the proton. So the isoelectric point, highlight this part. The isoelectric point is a pH. Okay, it's a pH at which particular protein carry no net charge anymore. Be careful, guys. It's no charge. You can say no charge. Okay, are you clear? So when the pH is higher than Pi, the protein tends to have a negatively charged. Okay. So always remember, if pH less than Pi, so protein will be positively charged. If the pH or vice versa more than Pi, so then it becomes negatively charged. Okay. So as pH solution increase, the deprotonations take place. Okay, therefore it changes the charge. So isoelectric focusing is a technique for separating the protein molecule by differences in their isoelectric point. So IF can be resolved the different protein with similar size that cannot be resolved on the standard SDS page. As I say that normal SDS page cannot do anything. Okay. So can you see that, guys? In this case, we have low pH, we connect to the positive terminal, high pH, we connect to the negative terminal, then they're going to move based on their isoelectric points. Okay. So this is the answer, you can try. Okay, then after that, you can check your answer. So try to get this correct. Try to get this correct. So in the case of hemoglobins, okay, this migrates to migrates to the zone in a medium where pH of the gel match with the hemoglobin isoelectric point. So at the Pi or isoelectric point, the charge of the hemoglobin becomes 0, the net charge, and cease to migrate. So if you look at this, hemoglobin A is a normal hemoglobin with 2 alpha, 2 beta, so about 97% in adults. So with isoelectric point 6.90. How about this? The S, hemoglobin S. Hemoglobin S actually also alpha 2, beta 2, but this one is for the sickle cell patient. Sickle cell patients. S. So you can see that in the normal adults, you won't have it, normal adults. But in the sickle cell traits, yes, they're elevated. And the Pi now is 7.28. Okay, 7.28. Then we have the fetal hemoglobin. It's very, very little in our body with 2 alpha and 2 gamma at Pi equal to 7. So if we subject these three together, you can see that they start to migrate. They start to migrate. So if you migrate, you can see that they migrate from positive low pH to the negative terminal, which is high pH. Are you clear? So predict what will happen if we carry out this gel electrophoresis based on isoelectric point. So under normal patients, or normal patient, under normal individual, we do know that normal individual will have the HbA and HbF. Oh, sorry. HbA and HbF. Are you clear? So where is the band? Look at this. Where is the band? So the bands, normal is 6.90. Fetal hemoglobin is 7.00. And sickle one is 7.28. So means that normal, they don't have sickle cell hemoglobin, right? So if no have sickle cell hemoglobin, then you can see that it forms 2 bands. One is at 6.90 Another one, will form at 7. Okay, I start with the normal. You can try with the sickle cell trait. Okay, so sickle cell traits. So sickle cell trait basically means that they will have HbA normal, they will have HbF, and also HbS. Not sickle cell trait, basically they are carrier. Okay, so 50% HbA, then they have 1% of HbF, another 50% will be HbS. Sickle cells, they don't have normal, but they have HbF and also the HbS. Can you see that? This is how we can separate the proteins based on isoelectric point. Okay, so no question, I stop the recording.